we should write here specific point, right? That is, in eukaryotes there is 7 mg cap. 7 mg cap is there on mRNA. This mRNA we know where this is monocystonic messenger RNA, right? On mRNA, and just complement complementary codes are present, complementary anticodes are present, anticode are present in where 18S ribosomal RNA of 40S smaller subunit. Of 80S ribosome. Do you understand? 80S ribosome means they have 40S small subunit. Small subunit is 40S, large subunit is 60S. So 40 subunit it is made up of 18S RNA. 18S RNA it has anticode of just 7 mg cap. So they recognize through this one and attached. So here we understand how a smaller subunit is attached with the messenger RNA, right? Now a complex is formed. When a smaller subunit is attached with messenger RNA, then a complex is formed. Complex means what? Complex means we only add the name and then write complex, simply, right? Here what happens? Because 30S subunit is attached with messenger RNA, we should write here 30S, right? 30S subunit, right? It's not RNA, I write 30S subunit plus transfer RNA. Which transfer RNA? Sorry, messenger RNA, right? It is messenger RNA. Messenger RNA, right? Now what complex is formed here? Do you tell me the name of the complex? Simply, here the complex is 30S mRNA. We can write here. It is 30S mRNA. 30S means 30S subunit mRNA complex is formed. Here IF3, right? And for the formation of this complex, GTP is used, because without energy no complex is formed, right? One here one engine that is ion is also used here, Mg plus 2. It is also, it facilitates this process, Mg plus 2, right? You understand? The formation of Mg cap. Now this one is the small subunit plus messenger RNA. Now what happens? This transfer RNA, right, which is activated or charged transfer RNA, it comes from cytoplasm and attach and then recognize this complex, this messenger RNA code, and attach with messenger RNA, right? This one is called initiation, right? Okay, now here one more point is there. How a transfer RNA attach with this? You should write this one, right? Now this 30S messenger RNA complex, right, which is formed here, 30S messenger RNA complex, right, it reacts with formyl methionine transfer RNA complex. That is because here why I said formyl methionine, here amino acyl, which you can also said that it is a means amino acyl transfer RNA complex, right? But we know here first code is, initiation code is AUG, and AUG is responsible for methionine, right? In bacteria it is not a methionine, it is formyl methionine, right? So here we should write here formyl methionine, right, transfer RNA, right? You understand why I said here that formyl methionine transfer RNA complex? Because it is the initiation codon. So here we should write here formyl methionine, right, transfer RNA complex plus this 30S mRNA complex, right? When we use that, when these two comes near and attached with one, with the help of, right here, IF2 IF3 Mg plus, right? With the help of initiation codons and also magnesium plus, they need also ATP for binding. ATP means here it is not ATP, it is GTP here used. They use GTP guanosine. So we should write here plus GTP. Now what is the structure is formed? A whole structure is formed in which this charged DNA RNA transfer RNA is also there, and 30S messenger RNA, 30S ribosome and messenger RNA. All these three are attached. Now a long statement is formed. It is called 30S mRNA formyl methionine, 30S mRNA formyl methionine transfer RNA complex is formed. Right? A very long structure. We can write here. What is given here? 30S. I should write here 30S. Then after mRNA. Now here from here formyl for 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 myl methionine then transfer RNA complex is formed. That complex is formed. Means here transfer RNA, 30S subunit, messenger RNA, three things are attached with one another, right? By using of charge, this one is called charged transfer RNA. This is our smaller subunit, and messenger RNA, right? Now what happens then? Then this large subunit, it attached with this complex, large subunit, right? Now the larger subunit, last point here, larger subunit, that is 50S. We know very well in prokaryotic cell 70S means one is 50S and another one is 30S. Large subunit 50S each attached with above means this one complex and a. Now a complete structure is formed. Complete translation apparatus is formed. Is formed. Okay. Is formed. Okay. This complete translation apparatus is formed. Here larger subunit is there. Larger subunit, it has two. Actually, sometimes it is in some books it is given it has 3 sides, but in our NCERT it is given 2 sides. One is called A site, another one is called P site. In Some books it is given 3 sites, A, P and E site, right? Okay, now here what I tell, okay, it takes some time, right? In larger subunit, what happens? It contains two sites, especially two sites, that is A and P site. That means A and P site. But in some cases, it is also contained, that is they said another site, that is E site, right? E site. E means what? Exit site. So exit site means what? From this side, and transfer RNA is released, right? A site means what? Another one is of P site. A site means amino acyl site. And P site means what? Peptidyl site. Amino acid site, amino acyl site and peptidyl site. It is actually DYM, right? Peptidyl, it is not. Peptidyl site. And E, already I'll tell you, E for exit site. So first what happens? The first transfer RNA, and this first transfer RNA we know because it is just complement to AUG, right? Just complement to AUG. AUG, it is a genetic code which is responsible for the attachment of methionine, right? Methionine only, right? So this methionine attached transfer RNA, the first transfer RNA attached with, look here, it is attached with the P site. At that time, A site is removed with P site. First, at that time, A site is empty. That means A site is empty, right? But after first, that means from second transfer RNA. That means all the, we can say here, from second or except first. That means all the tRNA attached with, they must be attached with A site except first transfer RNA. I think you understand here. The first transfer RNA, because at that time P site is empty, so it can and attach with the P sites. At that time A site is empty. Now the second transfer RNA, it comes and attach with the A site, and from second onward, second, third, fourth, when they come, so they first have to attach with the A sites. Then they transfer to P sites, and from P site to E site, and from E site they exit from the larger subunit. So these two sites, they are present in the larger subunit, right? This one is, I write here. When this transfer RNA it attach, that is first transfer RNA attached with P site, what happens? this process of polymerization of protein synthesis initiates, right? And thus the process of polymerization of amino acid, or we can say protein synthesis, initiates. Right? So this is the initiation process. How a protein, messenger RNA attached with the small subunit, and this, when a complex is formed, this complex is attached with the transfer RNA, and when transfer RNA is formed, that is what this complex is attached with the larger subunits. That is the main region or main platform of the protein synthesis. Okay. So then after initiation, second process is our trans-elongation. After initiation, it is elongation. Elongation means now one by one transfer RNA came to the A site, then they transfer to the P site, and from P site they exit. Right? When they come to the A site, that happens what happens? A P site already one transfer RNA is present with its amino acids. Another come to the P site. When they come too close to each other, then what happens? A peptide bond is formed between these two amino acids. Right? Now A site, the transfer RNA which is present at the P site, it release the amino acids. Right? When it release the amino acid, what happens? Peptide bond is already formed at A site. A site, it contains two amino acids. Right? Then what happens? This first amino acid, it come to the E site and release from the, means it leave the ribosome. Now this second one, it comes to the first, means A site, and now new transfer RNA comes and it attach with the A site. Right? Then after what happens? Peptide bond is formed between this second and third, and this amino acid, it is released from the second transfer RNA. Now second come to the exit site and remove from the ribosome. Now this, because it P is empty, now this amino acid with this transfer RNA, that is third transfer RNA, it come to the P site, right? And this A site means fourth transfer RNA comes. Then after what happens? This third, it left from the ribosome. Then fifth come, this fourth come to the P site and then remove. Fifth come to the P site and from exit site it remove. Sixth come, seventh come, and so on. This process is continuous, right? And a peptide bond is formed between the P site and A site of amino acids, and due to the peptide form, a long chain is formed. This process is continuous until the stop codon comes. When stop codon means in a messenger RNA, if UAA or UGA, or it is also, we can say here UAG. If anyone came there, we know very well these three codes, they have no transfer RNA. So due to the absence of transfer RNA, what happens? This chain is break, right? And also the messenger RNA is released from the smaller cell, right? That means what happens? The protein synthesis stops. That is called termination, and this is given here, right? I can draw a structure, and through this structure I try to explain. Here what happens? This one is our large subunit, right? And this side it is our small subunit. Okay. First case. Second, large subunit and small subunit. Large subunit and small subunit. Right? Same also, large subunit and small subunit. Now here, this one is our transfer RNA. This one is our messenger RNA. Right? Here codes are there. We can write here 1, 2, 3, 1, 2, 3. These are codes. 1, 2, 3, 1, 2, 3, 1, 2, 3. Now 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Here 1, 2, 3, 1, 2, 3, 1, 2, 3. Right? And so on 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. This. These are genetic codes. This is the messenger RNA. This one. This is the and this one larger subunit. It has two sides here with another two sides. This side it is A side and this is the P side. P and A side. P and A side. P and A side. Right? Two sides are there. And in this side, what is there? Here ribosomal RNA. Right? There also these are the two sides and these are new RNAs. RNAs are there, right? If you understand here what happens here, you can see there. First what happens, it is an amino acids, right? And this one is amino acids. Amino acid, right? Now here amino acid, double amino acids. This side amino acid, this side 1, 2, 3 amino acids are formed. Amino acid and here 1, 2, 3, 4 amino acids are formed. Right? Now first what happens? This is the code. They match this code. They start matching the code. 1, 2, 3, 1, 2, 3. Match the code. Right? So this one amino acid is there, one amino acid there. This is the P side. What happens when they come to closer? What happens? A bond is formed there. This is called peptide bond. Peptide bond. In the formation of peptide bond, ribozyme is used, right? And this peptide bond is formed between the COOH group of P site amino acid. Means it is formed between the we can say COOH group and NH2 group of, this is the P site amino acid, this is the A site amino acid. Means this peptide bond, it is formed, this one is peptide bond, is formed between the COOH group of P site amino acid and between the NH2 group of A site amino acids, right? Look here what happens, when this bond is formed, then what happens, it becomes empty and then it removes from this side. When it removes from this side, right, then what happens, a new, here a new is there with amino acid, it comes to that side, right? When it comes to that side, look here, a new is there, right, with amino acid, it comes to that side and this, it moves outside. Simple this process happens, this messenger RNA, now this, suppose that is A, U, G, second one is C, T, C, G, C, U, so many are there. AUG is there. This one AUG comes that side. Now this is the CTC. Now third is GCU, right? It is, suppose that this one is AT. Sorry, not T. It is AUA, right? That is GCU, AUA, so on. And this one is AUG. Second one is our CTC. Third is GCU, AUA. Right? You understand here what happens. First AUG, then it moves to AUG comes out. That one is removed out. That is one removed out. Right? Then CTC. Then second one is removed. Then GUC come to the P. This GUC it comes to the P side. Now next this transfer RNA it come to the A side. Right? Which is just anticode of. Which has just anticode of GCU. Anticode of GCU means what? You know, complementary means it contains which anticode? G means what? C means what? G, U means what? A. CGA. It comes to that side. This one is CGA. So this CGA means it is the anticode of GCU. So when GCU come to the P side, what happens? This CGA it recognize this GCU and attach to the A side, right? When it attached to the A side, then what happens? This CTC, the amino acid which is present in the CTC, this amino acids, it forms a peptide bond with which one? These amino acids. Amino acids means which amino acids? Which is attached with this 5' with the 3' end of transfer RNA, right? And a bond is formed here. This peptide bond is formed. Now this amino acid is removed. It is released from this transfer RNA, right? When it is empty, this transfer RNA it is empty, means it has no amino acid. Then what happens? It released from the exit site, right? Now here AUA is there. That means what? What complement is there? A means what? U means what? A, and A means what? U. So here we can write here. This is U, A, U. It recognized with this one. When this we can say messenger RNA is moving to this side, or this ribosome it is moving on messenger RNA. This process is called translocation. This process is called translocation of ribosome. We can say a ribosome moves along the messenger RNA from codon to codon, and one by one new amino acids are attached with peptide bond to each other, right? That means P site, amino acid of P site binds its COOH group with amino group of A side amino acid, right? And then what happens? A long chain is formed like this one, long chain of polypeptide. This one is called polypeptide chain, right? And it happens up to when UGA. UGA means no transfer RNA. UGA means what? For UGA, no transfer RNA is there. That means that what happens? This formation of chain is, that is, no transfer RNA for UGA, because it is called stopping. So it is called as stopping codon. So due to the absence of transfer RNA, any more transfer RNA, this process is terminated. Okay. In case of prokaryotic site, for elongation, we have also elongation means what? Elongation. We have factors. That factors are factor for elongation. Factor for elongation means what? It is EF1, EF. Elongation factor simply we can write here 2 or EF3 sometimes. Then same as to here, factor for termination. We have termination factor TF1, TF2 or TF3. In case of prokaryote, these are present in prokaryotic cell. Okay, so this is the total process of translation. I think you understand this one. This ribosome, okay, you understand this one is our 5' end, right? And this side it is 3'. This side 5', this side 3'. Do you understand? 5', 3', 5', 3'. You understand? Here, ribosome, it moves which direction? 5' to 3' direction on messenger RNA, or we can say translocation process. Translocation process, it happens from through 5' to 3' direction, right? Now you have any doubt is there? Okay, because here peptide bond is formed. One thing I tell you, ribozyme helps. Okay, ribozyme is also help. Another enzyme is also there. It is called peptidyl. Because peptide form, that is peptide bond is formed. It is called peptidyl transferase enzyme. Here we can write here peptidyl transferase enzyme and ribozyme. In case of this is 23S RNA. These two are responsible for the formation of peptide bond or the formation of protein synthesis. Look here, this here is vector for termination. I write here TF1, TF2. They are also called, because in our NCERT it is even releasing factor, so we can set them RF1 or RF2 or RF3. That is not a matter. It is terminating factor or it is releasing factor. Releasing factor means what? The protein which is formed here, or the peptide, polypeptide which is formed here, right? This one is released from that is ribosome, or we can say ribosome, because it is released from ribosome, so it is called as a releasing factor, right? So this is the total amount of translation, and I think you understand here. So this is the total step in translation: activation of amino acids, then charging of transfer RNA, right? Then translation. So translation means it starts from initiation, then elongation. Initiation, I write here initiation, how it initiate, right? And then after elongation and termination, I tell you in to this figure, right? And that is the total to this figure, but otherwise it becomes very long, right? So I think you understand this one. If you have any doubt, you can ask in your hand, right? After this one, okay, one thing is also there. You say how this ribosome moves from one side to another side. Here also a enzymatic process. It is enzymatic process, energetic process is there. Here, because translocation, this process is translocation process happens here. So which enzyme is there? Translocase enzyme. Simply you understand, we use ASE. ASE suffix is used for enzyme. Process plus ASE, that is enzyme. Biology means it is easy, very easy. You understand, right? Because translocation process happens here. So which enzyme is responsible for the translocation of this one ribosome? It is translocase enzyme. Okay. Now come to the NCERT. I think you write this one. Okay. Now second point is our here, because it is given terminating translation means terminating factor we can understand, releasing the complete polypeptide who is responsible here. Here we can say releasing factors are there, RF1, RF2, RF3, right? Now regulation of gene expression, how a gene expresses itself. How a gene expresses itself. Regulation of gene expression refers to a very broad term that may occur at various levels. Various level means here 4 levels are given. Okay. Which 4 levels are there? Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, because eukaryotes nuclear membrane is present. Eukaryotes means monocystonic messenger RNA is found. GCAP means processing is there, right? That's why in eukaryotes both are different. Prokaryotes means no processing. There is no nuclear membrane is there. And translation and transcription, these two process, sometime what happens, they happens simultaneously. Means same time. One side it is transcription starts, another transcription is going on, another side translation starts, right? But in eukaryotes, it is not happens, because eukaryotes means different nuclear membrane is there. So first, what happens? RNA is formed. This RNA, it is going into processing, splicing, right? Capping and tailing is there. Then after, it has to come out of the nucleus, because ribosomes are present outside the nucleus. So first, then after it comes out of the nucleus, then translation starts. So these are the levels. But what is there? Transcriptional level at the, that is transcription means what? Formation of messenger. That is formation of primary transcript. Primary transcript means at the time of the formation of messenger. Second, processing level, that is regulation of splicing. Splicing, processing, right? Splicing and then capping and tailing is there. Third, transport of messenger RNA from nucleus to cytoplasm, right? Because ribosomes, they are present in RER, it is present outside the nucleus. Then translational level. Translation level means what? At the time of protein synthesis. 4 levels are given here. First, transcription level at the time of, means at the stage of formation of messenger RNA. Second is given here, processing level. Splicing is G-capping, tailing and splicing intron when intron is removed, right? Third one is when messenger RNA is moved from nucleus to cytoplasm. And fourth one is our translation level, means at the time of formation of polypeptides. The genes in a cell are expressed to perform. Gene in a cell are expressed to perform a particular function or a set of functions, right? For example, if an enzyme called beta-galactosidase, example is given here, lactoperon enzyme, right? So this is for lactoperon model. Beta-galactosidase. Beta-galactosidase, this enzyme is formed through Z gene, that gene is Z, synthesized by E. coli, is used to catalyze the hydrolysis of disaccharide. Galactosidase. Galactosidase means what? ASE, lactose. It digests the disaccharide. Which disaccharide? It is lactose, right? So lactose sugar is broken down this way. Lactose sugar, it is a disaccharide which is made up of one galactose and one glucose molecule, right? Lactose into galactose and glucose, the bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy sources, they would no longer require the synthesis of the enzyme beta-galactosidase, right? What is here? If the bacteria do not have lactose around them, when lactose is not present around them, right, to be utilized for energy source, they would no longer require the synthesis of enzyme beta-galactosidase, because we know no lactose is there, then what is the need of lactosidase, right? Lactosidase is only responsible for the breaking down of glycosidic bond between glucose and galactose. No lactose, there is no use of beta-galactosidase. Therefore, in a simple term, it is the metabolic, physiologic or environmental condition. Metabolic we can say because broken down is there, anabolic reaction. Physiological, right, and environmental condition because this E. coli, they are present inside the lactose sugar that regulate the expression of gene. Which one? Metabolic, physiologic, environmental condition. They regulate the expression of genes. The development and the differentiation of embryo into adult organisms are also a result of coordinate regulation of expression of several set of genes. That is a single zygote. It forms a complex multicellular structure. It is also example of coordinate regulation of expression of different set of genes. In prokaryote, control of the rate of transcriptional initiation is the predominant site for control of gene expression. Prokaryotic means control of the rate of transcriptional initiation. Transcriptional initiation. Initiate means start. It start or it cannot start. Right? It is regulated at the transcriptional level. Where? In which? In prokaryotes. Control of rate of transcriptional initiation is the predominant site for control of gene expression. In the transcription unit, the activity of RNA polymerase at the given promoter, because RNA polymerase binds through promoter only, right, is in turn regulated by interaction with the accessory proteins. Right? Interaction with the accessory proteins. Interaction means what? Because here regulator genes are there. Regulatory genes, they form the either initiator or inhibitor. If it is inhibitor, that means what? Gene expression will stop. Right? These regulatory proteins can act both positively, activators, or negatively, we can say repressors. The accessibility of promoter region of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequence termed as operators. Right? These operators means these proteins, they can bind with operator. Right? When they bind with the operator, either they are responsible for the activate the process or they stop the process. If they activate, they are called as activators. If they stop, they are called as inhibitors. Right? Or repressors. The operator region is the adjacent to the promoter element in most operons, and in most cases the sequences of the operator bind a repressor protein. Most cases, right? Operator is bind with a repressor protein, means it stop or we can say it switch off the process. Each operon has its specific operator and specific repressor. For example, lac operator is present only in lac operon. Here lac means what? Lactose sugar. And it interacts specifically with lac repressor only. Right? Okay, we will read this lac operon model and different type of genes in our next lecture. Okay, so one time you read this translation, and if you have any doubt, you can ask. Thanks.